Well, hello. So, dancing. Almost everybody can imagine their personal experience of dancing. However, because different people have different experiences of dancing in different contexts, with different people, with different degrees of engagement and enjoyment, we would naturally anticipate that different people would bring to mind different mental images of dancing. Now, the prime interest of the current talk is in identifying whether we can determine these person-specific elements of imagination in recordings of brain data that is elicited during the imagination of events such as dancing. So why would this not be the case? Well, contemporary neuroimaging technology such as functional magnetic resonance imaging is a noisy measure. So whilst this has been very useful in identifying a network of brain regions that are reliably activated during episodic memory recollection or episodic simulation, and these include inferior parietal cortex, medial parietal cortex, hippocampal and parahippocampal regions, and medial prefrontal cortex. And more recently, different types of imaginary events have been shown to be discriminable in brain activity taken from across regions of this network. It still remains unclear whether fMRI can capture fine-grained person-specific imagery of similar events. So this would be as opposed to broadly homogeneous representations that are similar across individuals. So our overarching hypothesis here is that fMRI activation elicited when imagining personal experiences reflects person-specific information. So can we take brain activity recorded as people imagine themselves dancing or other events and identify which person's brain that activity came from? So how are we going to do this? We are going to scan people's brain activity as they imagine themselves in a pre-selected fixed set of events. And then we're going to re-represent this brain activity in a representational similarity space. So we see for this particular person that wedding and dancing occupy a similar position in this space, and we might suppose that possibly this person associates weddings with dancing. Now, if we do the same thing for a different participant, perhaps we find a different pattern. And here we see that this participant's representation of wedding is very close in proximity to their representation of bar. So we might suppose that this person likes taking advantage of the free bar at weddings. Now, of course, there will always be differences in fMRI similarity structure across participants. So how can we provide evidence that these are meaningful differences or just noise? So we're going to approach this by having participants verbally describe each of the personal imaginary events that they have brought to mind. And we are going to turn these verbal descriptions into a quantitative model that we will also represent in similarity space. And we would hope that this model will also capture meaningful similarities with participant 1 with weddings and dancing, and participant 2 with weddings and free drinks. So we are going to test whether personalized models can identify the corresponding participant from their fMRI data structure. So we're going to compare models derived from participant 1 to their own brain data and to other people's brain data, and we anticipate that the congruent match will be greater. So in a little more detail now, we recruited 26 participants who imagined their personal experiences of 20 pre-selected scenarios. So 3 of these scenarios are illustrated here, and each participant provided a brief sentence-length verbal description of each scenario. So here we have our first participant imagining themselves watching their daughter dance at the cultural center for the dancing scenario. And our second participant is busy dancing the hora. So then these participants underwent fMRI as they reimagined the scenarios when prompted by standardized cues. So this person is imagining their resting scenario, laying in bed in the morning, listening to the birds singing. Then to quantitatively model event descriptions, we borrowed a distributional semantic modeling approach from computational linguistics. So the basis of this is a model that captures generic representations of word level meaning that have been computationally induced, measured from the statistics of word usage in large text corpora such as Wikipedia. So under this approach, words are represented as multidimensional vectors, where each vector dimension reflects how often a modeled word co-occurred with other words across, say, Wikipedia. So words with similar meanings tend to appear in similar textual contexts, and because of this, they end up with similar vector representations. So to take these generic word level vectors forwards to model person-specific verbal event descriptions, we followed this approach. So we took each person's description of each scenario. We looked up the content words in that scenario description. We identified the semantic vectors that these content words correspond to, and we simply summed them together to build a person-specific representation of each scenario. So the second participant's resting scenario would be constructed from a summation of laying bed, early, beautiful morning, listening birds singing, and so on. We repeated the same procedure for each scenario and also for each participant. To compare model to brain data, we implemented a standard representational similarity analysis where both model and brain data were first re-represented in similarity space by computing inter-scenario correlation matrices. And this enabled models to be compared to brain data by computing Spearman correlation between the correlation matrix representations. So to test the hypothesis that fMRI data reflects individual differences in mental imagery, we took pairs of participants and computed RSA between congruent model and fMRI data pairings, so between participant 1's model and participant 1's brain, and participant 2's model and participant 2's brain, and compared these to the strength of RSA match between incongruent pairings, with the expectation, obviously, that congruent pairings would yield stronger RSA coefficients. And this was repeated for all 325 pairwise participant combinations. Turning to the results, so we conducted a battery of region of interest and searchlight-based analysis, which yielded the general consensus that medial parietal cortex reflects person-specific elements of mental imagery. We were able to discriminate individual identity in medial parietal cortex at an accuracy of around 75%, where our chance level discrimination would be 50%. In conclusion, this talk has provided evidence that fMRI activation elicited imagining personal experiences reflects person-specific information. We've seen that this effect appears to be particularly pronounced in medial parietal cortex. And at a more general level, we provided evidence that fMRI and computational models provide different ways to quantitatively measure episodic recollection or simulation at a personal level. Thank you for listening.
Hello everyone, thanks for joining my talk on real-time reconstruction of letter shapes in the mind's eye. My name is Rieck van Hoof and I'm a PhD candidate at Maastricht University. My supervisors are Dr. Mario Zende, Dr. Joel Reitler and Professor Rainer Goebel, who is the head of our Cognitive Neuroscience department. As this talk is pre-recorded, please save your questions for the end of the talk. Brain-computer interfaces, or BCIs, allow direct communication between a brain and external device. For people with severe motor disabilities, such as people with locked-in syndrome, you can imagine how having an alternative way to communicate has a dramatic effect on their life and the life of their loved ones. And even though a real-time fMRI-based BCI is relatively expensive, these people rely on technology on a day-to-day basis, and this research may lead to a new portable and patient-tailored BCI solution. In 2013, Nacy and Owen showed in a case study that patients diagnosed with being in a minimally conscious state or vegetative state were able to follow commands when asked to count, and able to communicate by focusing selective attention. So they were instructed to count if their answer to a question was yes, or to relax for no. And this is a way of encoding and decoding by respectively linking commands to a certain brain state and extracting distinct responses. Similarly, a study from our research group showed that any letter can be robustly decoded by means of mental imagery strategies, such as inner speech, mental calculation, and motor imagery. However, this is a rather indirect way of encoding because it recruits brain areas which are unrelated to the message. This can feel unnatural and cause high cognitive load during communication. We are therefore interested in a model which allows direct encoding, where brain activity directly corresponds to the encoded message. Luckily, parts of our brain are retinotopically organized and encode the visual field in a spatial cortical map. This encoding model is easily invertible with population receptive field models by projecting the retinotopic voxels back into the visual field. Last year we published a paper showing that indeed we can reconstruct visual percepts, and we took it a step further by reconstructing letter representations from the mind's eye. So the images of letters H, T, S, and C, as shown here in the bottom row, are retrieved from brain activity in the absence of visual stimulation. So our main question now is, can we find out what a person is thinking by reconstructing imagined images in real time? And to start answering this question, we acquired pilot data from 3 participants on our 7 Tesla scanner in Maastricht. Participants started with a retinotopic localizer showing flickering checkerboard bar stimuli, so we can calculate the location and size of the receptive fields for voxels located mainly in early visual cortex. In the real-time experiment, subjects are cued to imagine a letter shape, and while the subject is performing imagery, we invert the voxel pattern activity back into the visual field. Well, in this case, we mapped it onto a 150 by 150 pixel image and show the reconstructed image to the participant, forming a closed-loop system. Zooming in on the first step of the inverting process brings us to the population receptive field model. Since the classic PRF model uses grid search to find the PRF parameters, it requires too much computational time and resources for our purpose. Therefore, we use real-time retinotopy. This technique, developed by my colleagues, uses a tile hashing procedure to reduce the amount of data by 98% and estimates population receptive fields which closely resemble those computed by the classic model, but does this in a matter of seconds. For details on this beautiful work, please visit my colleague Salil Bhatt's poster with number 1512. So now we have for each voxel the location and the size of its corresponding receptive fields and can map it onto an image, as we see here in these average reconstructions of perceptual and imagery trials. Now the problem is that when you compare voxel patterns based on visual stimuli and imagery, is that the imagery voxel patterns are much weaker and noisier, as these patterns likely arise from top-down modulations. So to tackle this issue, we use an autoencoder neural network trained on single perceptual trials of an independent dataset. And by feeding the imagery reconstructions into the autoencoder, it effectively denoises the input image based on their geometric information. So for that to work, we added noise to the training examples to mimic and exaggerate a lower SNR. Here you can see the before and after denoising. For our real-time experiment, we want to pre-train the autoencoder for a subject that it has not seen before. And to make this possible, we need examples of many subjects. However, when combining voxel patterns across subjects, it is difficult to perfectly align that data. So we solve this by combining single trials in image space, instead of combining the MR data. So now we have the pre-trained autoencoder and the PRF estimates from a retinal topic mapping run and can start the BCI experiment. The participant imagines letter shapes for about 10 seconds, and based on the expected bold response in a select group of voxels, we reconstruct their mental image, denoise and segment it before presenting it on screen. And in parallel, we downsample the image and classify the reconstruction. Actually, we plan to acquire more data and expand to 4 letters before presenting our work here today. Unfortunately, the situation in the last couple of months made this impossible. So here are very preliminary results based on our pilot scans. This graph shows classification accuracy across 3 subjects who were trained to imagine the letters H and T. Each bar indicates a single run in which there were 10 trials. Participants seem to struggle in their first attempt. However, the overall accuracy is pretty remarkable, being around 80%. Moving on to our letter reconstructions, here's an example of a single run for subject 1. The top row shows raw imagery reconstruction trials, the middle row shows the correct letters, and the bottom row shows the denoised versions. As you can see, the raw reconstructions are simply unrecognizable. However, in most cases the neural network brings the image closer to a recognizable shape and provides feedback for the participant. So now for our second subject, you can see that from trial 1 to 10, every single letter was correctly decoded and reconstructed. So you can imagine the look on our faces when we witnessed the BCI was actually working. In conclusion, these first results are very promising. Naturally, our system has its limitations. Even with high field fMRI, we're still looking for the right trade-off between resolution and SNR and are subject to the sluggishness of the bold response. However, I think that we can carefully say that we succeeded to reconstruct the content of the mind's eye in real time. And we plan to expand this paradigm with more letters and participants, with the ultimate goal of creating a direct letter speller system. Last but not least, I'd like to thank everyone involved in this project, and our tools are made available for MATLAB and Python on GitHub. For more information on our work, please visit our group's website and our poster sessions. I'm now looking forward to your questions and thank you for your attention.
Hi everyone, I'm Lingxi from Peking University. Today the topic of my presentation is neural tracking of rhythmic constructs in imagined speech. Firstly, what is imagined speech, or we call speech mental imagery? Where mental imagery is a quasi-perceptual experience that can be internally represented without any external stimulation. For example, you may have the experience of like singing or speaking in your mind. And for speech mental imagery, it always requires the top-down regulation of memory retrieval in which the phonological, lexical, or semantic details of speech were rapidly organized and internally represented. Understanding how the brain constructs speech mental imagery is one of the most challenging problems in neuroscience. So in the past few years, there were many functional MR studies trying to localize the brain regions that are activated by imagined speech, and they have reported distributed neural networks, including the frontal cortex, like IFG, MFG, and the parental and the temporal cortex, as well as the premotor cortex and insular. They can be activated by different kinds of speech imagery tasks. However, we know that the low temporal resolution of fMR makes it is not the best way to characterize the rapid neural dynamics in imagined speech. And on the other hand, it remains highly challenging to track the neural dynamics in imagined speech because there is no external stimulation or behavioral output during the generation of speech mental imagery. So it is not easy to identify the imagery-related neural activities in milliseconds. To address this issue, we introduced the frequency tagging paradigm to capture the neural timescales in the frequency domain instead of the temporal domain. Frequency tagging is a promising approach for disentangling different cognitive processes when the corresponding neural activities change periodically at different frequencies. And recently, it has been used to characterize the higher level processes like the music rhythmic perception and the speech linguistic construction. And in one of our pilot studies, we separate two processes, the auditory perception and mental imagery, in a rhythmic inner counting task by frequency tagging. And in this study, we presented a sequence of pitons with the presentation rate of 4 Hz. And we instructed the participants to silently count numbers in their minds with every 5 numbers in a group. So the rhythmic construction of imagined numbers was tagged at 0.8 Hz. And as a result, we detected 2 significant spectral peaks at the imagery-related and stimulus-related frequencies. And in contrast, in the baseline condition with no mental imagery task, only the spectral peak at the stimulus rate frequency was detected. So in this study, we were able to track the mental operation of silent counting using frequency tagging. However, we know that imagined speech is more than just counting numbers. Speech processing include multiple processing stages, from the analysis of lower level acoustic features to the higher level like linguistic or semantic processing. And recently, a novel metric for tagging the rhythms of phrases and sentences has been developed, and it has been tested in speech perception and comprehension in 2016 in Ding's study. In this study, the bottom-up speech were concurrently passed into the hierarchically organized structures. Specifically, they made a connected speech with the syllables rate at 4 Hz, and every 2 syllables made a phrase. So phrases at 2 Hz, and every 4 syllables made a sentence. So sentences at 1 Hz. The MEG data then showed perfect spectral peaks at the tagged frequencies in the corresponding conditions. So this beautiful work reveals the higher order linguistic organization in speech perception. And at the moment, what is less clear is the top-down induction mechanism in imagined speech, which also contains the higher order structures, such as the syllables, words, phrases, and sentences. How are these higher order structures, the embedded structures, organized into imagined speech? And is there any shared or distinct neural mechanism between imagined and perceived speech. To answer these questions, we collected MEG data from 24 young adults under 3 conditions. In each condition, we presented a sequence of pure as the reference sound, and its presentation rate was 4 Hz. And then we chose some traditional Chinese poems as the speech stimuli, because in the Chinese poems, the sentence, every 5 syllables made a sentence. So the syllable level rate was fixed at 4 Hz, and the sentence level rate was 0.8 Hz. Then, in the imagery condition, participants were instructed to silently recite the poems in their minds, following the reference sound, without moving their lips or tongues. And in the perception condition, the actual poem was presented along with the reference sound, and the participants carefully listened to the speech stimuli. And in the control condition, participants silently counted the numbers in their mind without following the pace of the reference sound. And let's see the result, where firstly, we observed the significant spectral peaks at 0.8 Hz, corresponding to the signature rhythm in both imagery and perception conditions. And we also observed the significant responses at the stimulus rate frequency 4 Hz, which makes sense because we presented the sequence of pure tones, the reference sound, under each condition. And then we retrieve the response power at each tagged frequencies and compare it between conditions. And then we find similar response power between imagery and perception conditions at the signature rhythm frequency. And also, when we retrieve the waveforms in the time window of each sentence, we can observe the robust ERF, 5 ERF peaks during the time window of each sentences, which was induced by the 5 pure tones or syllables. And interestingly, we can observe the similar decreasing trend of the ERF peak amplitudes in imagery and the perception conditions, but not the control condition. So these results indicate that there are similar neural representations of the sentence level constructs in imagined and perceived speech. Then furthermore, we took advantage of the minimum L1 norm source estimation method at each tagged frequency bins. And firstly, we found extensive neural activation centering in bilateral auditory cortex at the stimulus rate 4 Hz, which also makes sense, and it might mostly reflect the auditory processing of the bottom-up auditory input. And also, we found 2 critical neural clusters activated at the signature rhythm in the imagery condition. They were left inferior frontal gyrus and the right supramarginal gyrus, with a shared cortical error in the right SMG relative to the activation map in the perception condition, and also some weaker activation in the left precentral gyrus at the sentence rate, and also at the left STG and MFG at the stimulus rate. So this source level activity also supports the neural representation of the higher order rhythmic constructs in imagined speech. Then taken together, by employing frequency tagging, we were able to track the rhythmic neural activities induced by the internal construction of sentences from syllables in both imagined and perceived speech. And we found similar neural responses to the signature rhythm in these two conditions. And also we reviewed the shared and distinct cohorts of cortical areas um, that were responsible to responsive to the tagged rhythms in these two conditions. And these findings demonstrate the top-down induction mechanism of the higher order rhythmic constructs in speech mental imagery. So I'd like to thank Professor Jiahong Gao and all the lab members in the Center for MR Research, Peking University. Thank you.
Hi. So auditory imagery is a common part of our daily lives. Whenever you imagine the happy birthday tune while washing your hands, you're likely to experience some auditory features of the melody in your mind without actually perceiving any auditory input. And music is one of the most complex auditory content people can imagine, often containing many auditory components that evolve over time, creating an engaging experience that resembles to some extent the experience when listening to music. So it's not surprising that studies showed a similar set of cortical regions that are active in both perception and imagery of music, including mostly secondary and associative auditory cortexes, as well as other frontal and parietal structures, such as the supplementary motor areas. However, most of these neuroimaging studies only identified a basic cortical overlap in the average signal across perception and imagery, and therefore could not detect potential differences across these two states in the neural processes that might take place in a region. For example, they cannot identify whether a region might contain information about a specific melody in mind, or be involved in a more global process that is not unique to the music content. So in this study, we asked to what extent do neural responses during imagery of musical sequences resemble the responses during perception. We wanted to know not just which regions are active in both perception and imagery, but whether they function in the same way across these two forms of musical experience. More specifically, we asked whether such a region might represent the specific musical content similarly across perception and imagery, or otherwise has a more general involvement in these processes that is not that is invariant to the specific auditory content. And finally, we know that the auditory system does not work in isolation when complex sounds are perceived. The neural representation of sound was shown to be modulated by movement, such as enhancement of perceived sound by rhythmic tapping. Therefore, we also asked whether movement could also modulate the representation of music during imagery. In each of our participants started the experiment by memorizing 6 melodies, each about a minute long, taken from soundtracks of animated movies. These instrumental melodies were matched for the timing of their beats and had no lyrics. Here's the beginning of one of them, for example. Participants learned independently to hum each melody very accurately, accompanied by a visual metronome in the form of a bouncing ball to guide their tempo. In the scanner for each melody, each participant first listened to the melody with an empty screen, then imagined the melody in silence twice, once with and once without finger tapping to the rhythm of the bouncing ball. We also had a control condition in which participants viewed the ball, but without the musical imagery task. Instead of the traditional task versus baseline comparisons of average level of ball, we used inter-subject comparisons analysis to compare the temporal pattern of the signal. And in this method, in each region, we correlate the full minute-long time course between different subjects while they, for example, imagine the same melody. This correlation value provides us a measurement of how reliable the signal is across people within that region when they imagine the melody. So when we see a reliable response across people, it means that the temporal alignment must come from an experience shared by them that synchronized their brain activity, which in this case would be the imagery of the melody. When there's no shared experience, for example, in a typical resting state, there's no reliable response across subjects. Using this method, we began by establishing to what extent and where in the brain imagining the same melody elicited synchronized neural responses across people. We performed the analysis within 400 parcels in the entire cortex, which were independently defined based on resting state. We found a reliable temporal response across participants at the center of the dorsal part of the superior temporal gyrus bilaterally, which largely overlap with facial gyrus, as well as in associative auditory regions in more anterior and posterior dorsal areas of the gyrus, and in lateral parts of the right SDG. For comparison, we performed the same analysis for the control condition when subject viewed the same metronome without performing the imagery task, and no reliable response was observed in this control condition. So this means that the common task of imagery was the factor that evoked similar temporal patterns across people in early and associative auditory cortexes. But what does this similarity of response represent? The shared experience of imagery has many components, so the neural similarity might have been evoked by a common replay of the perceptual experience of music, but it could also be a result of the shared responses that are idiosyncratic to imagery, that are not represented during perception. So to identify regions with shared neural representation across perception and imagery, we turn to compare the temporal patterns, not just between people, but also across the perception and imagery conditions. This allows us to identify regions that respond similarly when imagining a melody and when listening to it, and therefore this correlation represents the shared representation of the melody reinstated during imagery. These comparisons reveal that only a subset of the regions that were involved in imagery have a similar response as during perception, including bilateral early auditory regions and associative auditory region, mostly in the right STG, 5 parcels overall. But this consistent response across imagery and perception could still be related to either general cognitive trends common to both tasks, or could represent a reinstatement of the specific musical content with temporal patterns that are unique to the experience of each melody. To test for the specificity of these reinstated responses, we use permutation analysis that compare the temporal pattern similarity between matching melodies against the non-matching melodies across perception and imagery. We can say that the neural responses of the specific musical content is reinstated if the similarity of the matching melody is greater than the similarity between the non-matching melodies. So this analysis was performed within the 5 parcels found earlier and revealed that the right associative regions in the center and somewhat posterior parts of the lateral STG are showing greater within versus between melodies similarity. We also performed the classification accuracy analysis for these 2 parcels. In this analysis, participants were randomly split into 2 subgroups, and the average temporal pattern was compared across these 2 groups in all melody combinations. This classification accuracy was calculated as the proportion of melodies correctly identified out of the 6. The procedure was repeated 200 using 200 random combinations of the average and then average across groups. The classification accuracy was found to be significant in both of the parcels in the right lateral STG. So together, these results suggest that the associative auditory regions in the right STG contain melody-specific patterns that are reinstated from perception during imagery. And now that we found, this melody-specific neural representation during imagery, we try to explore whether it might be modified when imagery is accompanied by rhythmic tapping. So for this purpose, we repeated the inter-subject analysis, but now between perception and imagery condition that include tapping. You might notice that in this case, there are overall more areas in the auditory cortexes that show similar response across perception and imagery compared to imagery without tapping shown earlier. And within these 9 parcels of the SDG, we also repeated the same discriminability analysis, which now revealed that tapping extended the melody-specific response to further associative areas in the dorsal SDG bilaterally, in addition to the right lateral ones identified before, but not to early auditory cortexes. So to conclude, in this study, we were able to trace the neural responses associated with familiar musical segments and show the common ability to reactivate them at will when bringing to mind musical content. And our results showed that the temporal response pattern that were shared across people during perception of music were reactivated in a melody-specific manner during imagery in associative auditory regions in lateral parts of the right SDG. And in addition, when imagery was accompanied by tapping, the distinctive responses to the melodies were reinstated in additional bilateral associative auditory areas in the dorsal SDG, making the overall neural activation during imagery more similar to perception. So these findings suggest a common neural activation for complex and continuous auditory content in regions where encoded information is largely sensory, bridging across perceptual and imagined experiences. Thank you very much for listening and for all the funding agencies.
So I'd like to present to you an fMRI decoding study about the neural representation of number and how this relates to math ability. If you'd like to chat about this further, feel free to find me at my virtual poster. So humans are unique in their ability to work with number. Like many other animals, we can perceive of number in a non-symbolic format. For example, if we compare this group of 7 apples to 5 apples. But unlike other animals, we regularly interact with numerical information in a symbolic format. So for example, with Arabic digits or number words. However, debate continues on some of the basic issues surrounding the neural mechanisms that support our numerical thinking. For example, it's still unresolved whether the encoding of symbolic number is grounded in the more evolutionarily ancient non-symbolic number processing mechanism, is independent, or is perhaps linked earlier in development and then decoupled over time. So our first study question is regarding the encoding of number in different formats. In our study, we ask whether symbolic and non-symbolic representations of number share cortical patterns of activation. This would provide evidence that, at some level, the encoding of specific numbers is shared across formats. Now, humans are also unique in the ways that we use number. Sometimes we're simply identifying a position or a place, like with platform 9 3/4 or number 10 Downing Street, but we're often acting on the numerical information, like when you're trying to figure out whether or not buying the organic version of that apple is worth it. So this brings us to our second study question. Are patterns of activity for number shared across tasks? In particular, we will investigate the contexts of identification and comparison. Now, while the basic science of these questions alone is interesting, they're also important behaviors in a broader context because basic numerical processing consistently relates to a range of mathematical skills. One of the most common number processing tasks is the number comparison task. Here, the participant simply chooses which of 2 numbers is greater, and meta-analyses indicate that performance in this task, in both formats, correlates with math skills. And while this is true across the literature, we don't really understand the mechanisms underlying this relation. So our third study question, we probe this relation by asking if cortical patterns in response to number relate to math skills. Our sample consists of 39 undergraduates with complete data that are about 20 years old. We use 2 fMRI tasks. In the identified task, participants are asked to identify with a button press whether the stimulus is a 2, 4, 6, or 8. And you can see some exemplars here on the right. In the compare task, participants determine if the number they saw was less than or greater than 5. It was an event-related design. Symbolic and non-symbolic stimuli were intermixed, and all of the stimuli were matched visually for average surface area of black pixels. We conducted our MVPA in 6 regions of interest. First, most research to date has pointed to the bilateral parietal lobes as the primary location for encoding numerical magnitudes. Second, Meta-analyses and cell recordings of homologous structures in macaques indicate that the inferior frontal gyrus encodes specific numerosities. And third, recent research into processing numerals in the ventral visual stream has indicated that there may be cortex specialized for number representation in the ventral occipitotemporal lobe, so frequently referred to as the number form area. So this is a spherical ROI generated from our recent meta-analysis on the topic. And lastly, our measure of math ability was a composite of the arithmetic fluency and calculation subtests of the Woodcock-Johnson 3. So a few details about our MVPA analysis and MRI data. We used the 7T Phillips scanner at Vanderbilt with a resolution of 2.5 millimeters. For decoding, we use a leave 3 out cross-validation technique. We use an LDA classifier implemented in Cosmo, and for all of our analyses, chance is calculated at 25%. Now, before we can answer any of our study questions about format comparison or task independence, we must first check to see if in our tasks and in our data, we have neural activity that relates to specific numerosities at all. And if you're unfamiliar with MVPA, essentially we're training an algorithm to identify spatial patterns and bold response to see if those patterns relate to conditions of interest. In our study, those conditions are the numerosities 2, 4, 6, and 8. So let's start with the non-symbolic numerosities. Now here's a confusion matrix from our results, where on the y-axis we have the target values, which represent the true conditions of the data. And here on the x-axis, they represent what the algorithm predicted. So here you have, you can see that when the actual data was at 2, the LDA classifier predicted 2 about 40% of the time, which is well above chance. And if you average over the diagonal, which is all of the correct predictions, then you get a sense for how successful, on average, the algorithm is across numbers. And the mean here is 36.8%, which is well above chance, or in other words, you can decode the numerosities. And then we want this information for each of our conditions and each of our ROIs. And when we did that, we found above chance decoding in all conditions in all regions except for the right NFA. Okay, so let's turn to our first study question. Do symbolic and non-symbolic representations of number share cortical patterns of activation? In order to answer this question, rather than simply training the algorithm on some of the data and testing it on another portion of the data, we train it to classify numerosities in one format, and then we test whether it can decode numerosities in the other format. Or in other words, can it generalize to the other format? If it can classify at above chance accuracy rates, then there's information in the pattern of neural activity that generalizes from one format to the other. And we take this to mean that at least some numerosity-specific neural resources are shared between formats. And when we look across our 6 ROIs, we see above chance decoding for both parietal regions and the left IFG, indicating shared neural resources in these 3 regions. Okay, now we move on to the second study question regarding task independence. And here we apply the same generalization testing procedure, except we train on the numerosities in one task and test on the other task. And when we look across our 6 ROIs, we see above chance decoding for both parietal regions and the bilateral IFG. So in answer to our question, yes, cortical patterns are shared for numbers across tasks in these 4 regions. And now for our last study question, do cortical patterns relate to math? Now, mathematics is almost entirely built on a symbolic number system. So one thing we're specifically interested in is whether generalization between formats relates to math ability. If we take a look back at our format generalization plots, we can see that there's quite a bit of variability, with some individuals showing better generalization, which may look like this example at the top, and some worse that may look like this example at the bottom. And when we ran this correlation, we found that individuals with worse generalization had higher math scores. And this was true for both the left and the right parietal ROIs. So this may indicate that people with more distinct format representations are better at math. Now, I find this doubly interesting because there was a finding a couple of years ago that showed nearly the same correlation with a similar measure of math achievement. So essentially, we replicate this finding. So to review our findings, we did find evidence for shared neural resources between symbolic and non-symbolic number formats in the identify and compare tasks in the parietal lobes and IFG. So broadly, we're confirming that these regions encode number such that it is recycled across different ways of using number. Here, I think that the biggest unresolved questions are developmental in nature. So for example, how do we acquire symbolic number, and what specific relation does it have to the pre-existing non-symbolic representations that we share with other animals? We also showed that generalization between symbolic and non-symbolic formats negatively predicts math achievement, in line with previous reports. So what does this mean? Well, several research groups have theorized that symbolic and non-symbolic number are related early in development when children acquire symbolic number, but that symbolic numbers are decoupled as they take on more properties of exact symbolic number systems. This negative correlation could indicate that there are individual differences in the degree of that decoupling that relate to math achievement in adulthood. But this is really a story that needs further empirical evidence. I'd like to thank my collaborators and our funding sources, including my postdoc funding from INSERC and BrainScan. And if you're interested in the topic of number representation, we have another study that just came out in Network Neuroscience that takes a graph theory-driven approach at network organization while processing symbolic and non-symbolic number. Here's the title and DOI, so feel free to take a screenshot. Thank you.
Hi, everybody. Thanks for joining me today. Today I'll be presenting my work looking at brain structure and function to predict different aspects of cognitive control in a normal, healthy aging sample. Cognitive control refers to the cognitive mechanisms that modulate and oversee the flexible allocation of mental resources during some kind of goal-directed behavior. So for example, if you were in the kitchen with the goal of cooking dinner, you might have to keep in mind all the ingredients that you need for the dish, when to execute each step of the recipe, and then update your priorities based on how things are going. You might also be switching between two different tasks, like sauteing meat and chopping vegetables. All the while, you might also be dealing with interfering or distracting stimuli like a hungry pet that's begging for scraps. So you can see that cognitive control encompasses a wide variety of processes underlying this broader complex goal. In the lab, cognitive control is considered as a more general construct that can be broken down into correlated but distinct domains that each have a unique contribution. One of which is inhibition, which refers to the controlled suppression of a predominant or interfering response. Another is shifting, which involves being able to flexibly switch between task sets or task rules. And finally, working memory, which involves updating and monitoring stored information through some kind of active manipulation. When we look at what brain regions subserve different aspects of cognitive control, we do see a wide set of regions activated for any one given task. But when we just focus on those regions activated consistently across inhibition, shifting, and working memory, highlighted in green here, we see that there is a common core set of regions activated across these domains. Specifically, it's a set of frontoparietal regions suggesting some kind of a superordinate cognitive control network supporting a broad range of executive functions. In normal healthy aging, we do see declines in cognitive control across all domains. Normal aging is also characterized by degradation of frontoparietal gray and white matter structures, which may account for these cognitive control deficits that we see in aging. Similarly, studies examining functional activity during cognitive control tasks do find age-related alterations in functional activity in a variety of regions, including those in the frontoparietal regions of the brain. But it's still unclear how individual differences in structure and function might be interacting to explain differences in cognitive control, specifically for the individual subdomains. So the goal of the current study was to integrate behavioral, structural, and functional neuroimaging measures in order to examine both age and individual differences in structure-function relationships for each domain of cognitive control and the subsequent impact on task performance. We recruited 140 healthy, normal aging adults from the Toronto area, ages 20 to 86, to be part of a comprehensive neuroimaging study that included both gray and white matter structural scans, as well as functional scans associated with each of the 3 subdomains of cognitive control. To look at inhibition, we used a go-no-go paradigm in which participants had to rapidly respond to frequent X stimuli and withhold response to less frequent non-X stimuli. To examine shifting, we used a local switching task in which participants made judgments about whether a letter was lower or uppercase or a vowel or consonant. And for some of the trials, they repeated the same kind of judgment, whereas others involved switching. And finally, for working memory, we used a standard N-back paradigm with 0, 1, and 2 back working memory loads. For our structural measures, we used FreeSurfer to parcellate the gray matter cortical ribbon in our high-res anatomical scans, and we computed a composite of cortical thickness for frontal and parietal regions of the brain that have been highlighted here. We also collected diffusion-weighted imaging to examine white matter microstructure, and we used Tracula to isolate major white matter bundles connecting frontal and parietal regions of the brain, and extracted fractional anisotropy as our primary metric. For both the gray and white matter metrics, we also extracted thickness and fractional anisotropy from control regions to just better determine how regionally specific any structure function findings were. So in order to integrate all of these complex data, we used a multivariate approach, partial least squares, which allowed us to examine how age, in-scanner task performance, and frontal parietal gray and white matter structure correlated with patterns of functional activity associated with each task that we had in the scanner. And we did this separately by task and domain. And because we know that aging has an effect on behavioral performance and frontal parietal structure, we regressed age out of those measures in order to understand their contributions to functional activity independent of aging. In today's talk, I'm just going to focus on the partial least squares results, but if you're interested in the age effects on each of these measures, you can come visit my poster number 648. The inhibition partial least squares resulted in a latent variable of functional activity characterizing activity for no-go versus go trials, largely in middle frontal, medial superior frontal, angular precuneus regions of the brain that somewhat overlap with dorsal portions of the default mode. And when we look at the multivariate correlations with this pattern of activity, we find that decreased activity for no-go trials in these regions was associated with older age. And decreased activity was also associated with greater fractional anisotropy and frontoparietal white matter, and this was independent of the effect of age. And importantly, this pattern of activity did not correlate with white matter fractional anisotropy in our control pathway, the corticospinal path, suggesting that this relationship was somewhat specific to frontal parietal white matter. And finally, we find that increased activity in this set of regions was associated with a faster speed accuracy trade-off on the go-no-go task. And again, this was independent of age. We found no structure function associations with frontoparietal thickness on the inhibition task. The switching partial least squares identified a pattern of activity associated with switch versus repeat trials in a broad set of regions in the brain, including middle and anterior cingulate, occipital, anterior temporal, and orbital frontal. And here we see that increased activity to switch trials in these regions was associated with older age, whereas decreased activity associated with switch trials was associated with greater frontoparietal cortical thickness after controlling for the effect of age. And again, this pattern of functional activity did not correlate with thickness in our control calcarine region, suggesting again that it was somewhat specific to frontoparietal thickness. And there were no structure function associations for white matter or task performance for the shifting analysis. Then finally, the working memory partial least squares resulted in a fairly restricted pattern of functional activity in left middle temporal gyrus and superior supplementary motor, and middle cingulate that was associated with activity for 1 and 2 back working memory loads versus 0 back. And this pattern of activity, decreased activity in these regions during 1 and 2 back trials was associated with older age. And decreased activity was also associated with better accuracy for 1 and 2 back trials. And there were no structure function associations with either gray or white matter for the working memory analysis. So together, these results show that age, behavior, and brain structure have unique contributions to functional activity, largely in default regions of the brain, but this tends to differ by cognitive control, specific cognitive control domain. When comparing across tasks, we see that aging is associated with overall decreased activity for inhibition and working memory, but increased activity for shifting. Furthermore, independent of the effects of age, and scanner task performance on inhibition and working memory is supported by individual differences in functional activity. We also see that inhibition and shifting showed greater contributions of structure to overall decreases in brain activity. This was selective to frontoparietal structures, suggesting that intact frontoparietal gray and white matter might allow for the efficient use of functional resources and not needing to activate as much. And then finally, working memory showed no contribution of structure to functional activity, but did have the strongest age effect. Therefore, the aging processes might have a stronger influence on the neural correlates of working memory than just individual differences in structural integrity alone. With that, I would like to thank Cheryl Grady, my mentor, as well as past and present members of the Grady Lab, our funding resources, as well as you for your attention. And if you would like to see more, you can come visit my poster, number 648. Thank you.